So, welcome to this class on neuroscience of human movement. This is part 3 of our discussion on cerebellum. In this class, we will be discussing the details of climbing and massy fiber inputs and how they encode different types of inputs or different kinds of inputs into the cerebellum, and how excitation and inhibition within the cerebellum are compared by multiple parallel pathways and what are the various recurrent loops. In other words, we are interested in this class to discuss the details of the microcircuit of the cerebellum. It turns out that the cerebellum has thousands of repeating units of these microcircuits. So, it is the same fundamental unit that gets repeated multiple times within the cerebellum. Unlike other regions that we discussed earlier, for example, we discussed the case of primary motor cortex, premotor cortex, etc., where we said that the situation is that the representations are more mosaic and there is both convergence and divergence, and are regions that control a particular part of the body are located over a relatively large area in the motor cortex, etc., we discussed earlier. Unlike that, here what you have is a relatively regular arrangement. The same microcircuit has been repeated multiple times. So, that gives rise to questions that what could be the function of this microcircuit. So, how this microcircuit and its output and its function modulate output of the cerebellum as a whole and its function as a whole. So, those are the questions. So, in this class we will discuss microcircuit of the cerebellum and the details. It turns out that the delight in this sort of science is in the details, is it not? So, we will start out with the climbing fibers and massy fiber system. So, we said what are massy fibers. Massy fibers are, I will write this here. So, massy fibers arise from, these are sensory, massy fibers are sensory information, or massy fibers are neurons that bring sensory information from spinal cord and other regions of the CNS, and they also bring information from cerebral cortex via pons, right. So, they basically bring information from pons and sensory information from spinal cord and other regions, and some others not discussed as detail, but classified as others, right. These are the major inputs to the cerebellum, or at least one major input to the cerebellum. There are two major inputs to the cerebellum. These are coming from two different types of fibers. One is massy fiber, the other is climbing fiber. Okay. So, massy fibers form one major input to the cerebellum and they innervate with granule cells, right. We showed in a previous class that they innervate with granule cells and with Golgi cells in a glomerulus. We saw that in the previous class, is it not? So, it turns out that each granule cell receives input from a few massy fibers, but one massy fiber innervates several thousand granule cells. One massy fiber thousands of granule cells. So, one massy fiber can serve as input to thousands of granule cells, and I said that makes sense, right, because granule cells are very large in number, massy fibers are much smaller in number. So, there must be a great amount of divergence from the massy granule system, right, or the massy parallel fiber system encodes basically a great amount of divergence, involves a great amount of divergence from one mossy fiber to thousands of granule cells, right. So, there is divergence. This is the molecular layer, that is the Purkinje layer, whereas this is the granule cell layer. In the molecular layer, the axons of the granule cells become what are called as parallel fibers, and they innervate anywhere between 250,000 to 1,000,000 connections with the Purkinje cells. We have seen that earlier. So, in the previous class we showed that the Purkinje cell has this unique, very distinct dendritic tree as shown here, right. We said that there is thickness in the anterior posterior direction. Actually, it might look like mediolateral in this picture, but I am showing for the sake of illustration that these two directions in this paper, in this page, are anterior posterior direction, but this is very thin in the mediolateral direction. This is something that we discussed in the previous class. But what also happens is that the parallel fibers are perpendicular, run perpendicular to the dendritic trees of the Purkinje cells. Now, consider that each of these pages is a Purkinje cell, for example. So, here is a Purkinje cell and I have several such papers, several such pages, each of them is a Purkinje cell, is it not? So, that is a Purkinje cell, so that is another Purkinje cell, etc. Now, it turns out that they have great amount of territory, they cover great amount of territory in the anterior posterior direction, but each Purkinje cell is very thin in the mediolateral direction, and it turns out that the parallel fibers. So, these cables that I am showing here are the equivalents of the parallel fibers, right. So, the parallel fibers run primarily in the mediolateral direction, in that direction, for example, right. So, in this direction. So, that means they are perpendicular, to the plane of the Purkinje cells. In other words, they have the potential to make connections with all these Purkinje cells, and several of these. So, what I have shown is only like 3 or 4 parallel fibers, but we said the number of parallel fibers are 100 billion, is it not? That is a very large number. So, these parallel fibers run perpendicular to the plane of the paper like that, in that, and make connections, make synapses with these Purkinje cells. So, okay. So, we discussed how Purkinje cells get inputs from thousands and thousands of parallel fibers or granule cell axons, right. So, another major form of input to the Purkinje cells is the climbing fibers. Where do they originate? We said where the mossy fibers originate. The climbing fibers originate from the inferior olive, right. So, these are special in the sense that these neurons or these fibers do not make connections with granule cells at the granule cell layer. They climb through, that is the reason they are called as climbing fibers. They climb through the cerebellar cortex, through the granule cell layer, through the Purkinje cell layer, and directly into the molecular layer, where the Purkinje fiber dendrites are there, and then they make connections with several dendrites of the Purkinje cell, fundamentally different from the way the granule cells function, the parallel fiber system functions. Let us remember that one mossy fiber can innervate about thousands of granule cells, and the number of inputs received by each Purkinje cell from parallel fibers can be of the order of 250,000 to 1,000,000. So, one Purkinje cell receives input from about 1 million parallel fibers, or 250,000 to 1 million, very large number of inputs are received by one Purkinje cell, whereas one climbing fiber innervates about 1 to 10 Purkinje cells. So, one climbing fiber can activate about 1 to 10 Purkinje cells, but a given Purkinje cell receives input from only one climbing fiber. What do I mean by that? So, let me repeat that for clarity. So, one climbing fiber innervates, suppose there is this climbing fiber, this innervates this Purkinje cell, and let us say there is one more Purkinje cell here whose dendritic tree is here. So, this Purkinje cell then branches out and also innervates these Purkinje cell dendrites, and also one more Purkinje cell. So, that is the climbing fiber to Purkinje cell connections, right. But this Purkinje cell, I am going to call that as cell 1, it receives input only from climbing fiber 1, whereas there might be a different climbing fiber that I am going to now draw in blue for the sake of clarity. This climbing fiber innervates a different Purkinje cell, that I am going to draw in a different color for the sake of clarity, right. So, that climbing fiber continues and innervates these. So, cell 1 is going to receive input from say for example climbing fiber 1, and say this is cell 2 that is receiving input from climbing fiber 1, whereas the climbing fiber shown in blue is climbing fiber 2 that is innervating cell 3 or 4. So, I am going to call this as cell 4, that is innervating cell 4. So, a given cell, a given Purkinje cell, say PC1 or Purkinje cell 1 is receiving input from only one climbing fiber, either from climbing fiber 1 or from climbing fiber 2, but climbing fiber 1 itself can innervate multiple Purkinje cells. Okay. So, that can be of the order of 1:10. So, one climbing fiber can innervate multiple Purkinje cells, but a given Purkinje cell can receive input from only one climbing fiber. So, it seems like there is a fundamental difference in the way these systems function. What you have at the mossy parallel fiber Purkinje cell system, what you are having first is a great amount of divergence. One mossy cell can innervate thousands of granule cells, and hundreds of thousands of granule cells or parallel fibers innervate a Purkinje cell. So, 100,000 for example parallel fibers innervate a Purkinje cell. So, what is this? This is convergence, is it not? So, you have this great divergence convergence of inputs from the mossy system, whereas in the climbing fiber system you do not have this, you have direct relatively direct inputs, and by the way the inputs that are given by the climbing fibers are fundamentally different when compared with the inputs of the mossy system, right, or the mossy granule cell system. Also important to note is that the climbing fibers that arise from similar regions of the inferior olive project to a relatively small area. So, there is topography. So, climbing fibers arising from similar areas in the inferior olive project to a relatively small area in the cerebellar cortex, and the Purkinje cells that receive inputs from this region also project to a relatively small area in the deep cerebellar nuclei. We are not done with the discussion of the microcircuits, there is more. Actually, what is the nature of these connections? What is the nature? Are these inhibitory or excitatory? Are things that we should start discussing. It turns out that the mossy granule cell system is an excitatory connection. Its input is excitatory, its its neurotransmitter is glutamate. Right. And the climbing fiber Purkinje cell system is also excitatory. Its neurotransmitter is also glutamate. However, there is a difference in the way these function, that detail we will discuss, and the granule cells also excite the Purkinje cells, whereas the Purkinje cells actually their outputs are inhibitory. The output of the Purkinje cells are basically governed by GABA, inhibitory. What is not shown are other details that are shown in, for example, in this picture, right. So, it turns out that there is more to this than the what was shown previously. So, for example, the climbing fiber that originates in the inferior olive proceeds and actually innervates the Purkinje cell, actually makes thousands and thousands of synapses with the Purkinje cell dendrites, right. But they also branch out and excite the deep cerebellar nuclei neurons. Neurons in the deep cerebellar nuclei are also excited by the climbing fiber system. But the Purkinje cell is actually inhibitory. So, what is happening is a comparison of excitation and inhibition, that is probably what is being performed, is it not. So, the Purkinje cell inhibits the deep cerebellar nuclei, right. So, that forms a recurrent loop that is shown here, right. So, there is excitation that travels in this direction, and then there is inhibition that travels down into the deep cerebellar nuclei from the Purkinje cells. But it is not just that, it turns out that what is also true is that the deep cerebellar nuclei can also modulate the functions of the inferior olive. This is inferior olivary nucleus. Okay. So, the deep cerebellar nuclei can also modulate the functions of the inferior olive by inhibiting them. By the way, what is also what is not mentioned previously is that the inferior olive, right, it is special in the sense that within the brain, among the excitable cells, there are a few cells that conduct electrotonically. We discussed previously, communication between cells happens in two manners. Basically, synapses are of two types. One is electrical synapses and the other is chemical synapses, is it not. Mostly we have discussed chemical synapses, which are the most prevalent, but there are some cases where the synapses are electrical or the connections are rather electrotonic. That kind of activity, that kind of phenomenon happens in the inferior olive, where cells in the inferior olive communicate with other cells in the inferior olive through what are called as gap junctions. Very special. These are also found in cardiac cells, for example, but that is outside our discussion. So, within the inferior olive, so there are these gap junctions and electrotonic coupling between this. So, it turns out that it is because of electrotonic coupling. If one climbing fiber fires, all the climbing fibers that are electrotonically coupled to it fire, leading to relatively synchronous activity of the climbing fibers. Okay. So, and because deep cerebellar nuclei can control the extent to which the activation comes from the inferior olive, in a way modulation of cerebellar activity is performed by the cerebellum. Cerebellum within the deep cerebellar nuclei within the cerebellum controls to what extent it is going to be excited or inhibited. So, to some extent that is controlled by the deep cerebellar nuclei here. But there is one more loop, right. The Marcy system excites the granule cells, and they also excite, the Marcy system also excites the Golgi cells, right. What happens is that the granule cells excite the Purkinje cells, which further inhibit the deep cerebellar nuclei, but it turns out that the Marcy system also causes an excitation of the deep cerebellar nucleus. So, once again what is happening is an excitation is first given to the deep cerebellar nuclei, and that excitation is carried to the Purkinje cell layer, which then causes an inhibition. The output of the Purkinje cells is always inhibitory. So, that causes an inhibition. So, at this stage what is happening is a comparison of that excitation and that inhibition at that level, and also a comparison of that excitation and that inhibition, right. So, there is this loop. But what is not discussed is the case of Golgi cells. Marcy fibers also excite Golgi cells, and these Golgi cells inhibit, Golgi cells are inhibitory interneurons, they inhibit the granule cells. So, there is a loop here. Okay. We discussed the case of the climbing fibers and Marcy fibers, what are and the output from the Purkinje cell, this is always inhibitory. What we have not discussed is the case of inhibitory interneurons, other inhibitory interneurons. There are two major inhibitory interneurons in the molecular layer. These are basket cells and stellate cells, right. Stellate cells are star shaped cells, that is the reason they are called as stellate cells. These have relatively short axons and they cause inhibition of nearby dendrites of the Purkinje cell. They are excited by the parallel fibers. So, they receive excitatory inputs from parallel fibers and they inhibit nearby dendrites of the Purkinje cells. Whereas, basket cells are unique, they also receive inputs from parallel fibers, but they project onto the Purkinje cells, sometimes to the cell bodies of the Purkinje cells and nearby dendrites of the Purkinje cells, causing relatively strong inhibition when compared with the stellate cells. The inhibition caused by the basket cells is relatively strong, and they function. So, they function in such a way that when there are, so let us remember that there are going to be thousands and thousands of parallel fibers, right. So, the basket cells function in cause inhibition in such a way that only some of these inputs are going to actually cause only some of the inputs are going to actually cause excitation, while ensuring that the remaining ones continue to be inhibitory. So, these are inhibitory, these are so are their effects are controlled or modulated in such a way that among thousands of parallel fibers, a central flank alone will cause an excitation, whereas the others will not cause that, and that act is not on the parallel fibers, but rather on the Purkinje cell. So, in a way it is functioning to cause focus or sharpening of the parallel fiber input. Basket cells function to focus or sharpen the outputs of the Purkinje system. So, different functions by both are inhibitory interneurons, both stellate and basket cells are inhibitory interneurons, but their functions are different. right. So, what are the other things? There are other cells whose functions are not discussed in this class. Excitation inhibition balance that happens at multiple levels, right. First, there is excitation of the deep cerebellar nucleus by the inputs, namely the Marcy system and the climbing fiber system, and these are whereas the deep cerebellar nuclei are inhibited by the outputs of the Purkinje cells, and the Purkinje cells themselves are excited by the climbing fiber and the Marcy parallel fiber system, right. And the output of the Purkinje cell is modulated by inhibitory information that is received from the stellate and basket cells. Okay. This is what we have discussed so far, and what else are there? What is the special nature of the climbing fiber and its output is something that we must discuss, right. The climbing fiber produces what are called as complex spikes. What are these? It turns out that the climbing fiber causes a relatively large amplitude action potential in the Purkinje cell, and then several bursts of several high frequency bursts of relatively smaller amplitude action potentials. This is because of protracted time. This is because of protracted voltage gated calcium conductance. So, the calcium conductance fundamentally different from the previous discussions that we have had. In this case, what happens is the climbing fibers cause calcium conductance for a relatively long time to persist in the Purkinje cells, causing these, right. The action potentials are going to be like this, right. This is called as a complex spike. Okay. This is different from the action potential that is going to be caused by a parallel fiber. The parallel fiber is going to cause a regular action potential. So, that are seen as these, right. So, at rest what happens? At rest, when a person is at rest, spontaneous activity of climbing fibers causes anywhere between 1 and 3 complex spikes per second. So, even when you are at rest, when you are sitting quietly not doing any activity, the climbing fiber causes anywhere between 1 and 3 complex spikes per second, whereas the activity of the Marcy parallel fiber system causes simple spikes at rest, sometimes even up to 100 spikes per second. The results of this action on the Purkinje cells are fundamentally different, because one is causing a complex spike or a relatively large amplitude output followed by several smaller amplitude action potentials. It is not clear whether the smaller amplitude action potentials of the complex spike are actually communicative, are actually used in the communication down the Purkinje cells. But note, even at rest the simple spikes can be as high as 100 spikes per second. What happens during activity? During activity, climbing fibers, so this is at rest. During activity, climbing fibers produce activity that temporally codes actions or temporally codes events. So, temporal coding of events by the climbing fibers, whereas the activity that happens due to simple spikes, due to the Marcy parallel fiber system, can cause up to several hundred spikes per second. So, that activity is relatively of high frequency So, also note, so this means it seems to us that the difference that the climbing fiber and mossy fibers fundamentally code different things. It seems like that the climbing fiber system is more of a presence or absence switch, or it acts more like a signal for the start or end of an event, right. Whereas, the activity of the mossy system in some sense encodes the amplitude or the extent to which the activity is happening. So, one is a start stop switch, it can be considered, and that the climbing fiber acts more like an event signal or presence or absence of an event, whereas the mossy system encodes the actual amplitude of the event. There is more to this. This is a very simple description of what is actually happening, and the actual situation is way more complicated. I have made an attempt to explain this in some terms, at least we should make an attempt to understand this at some level. So, I have not done justice to the complexity of the cerebellum here, that is not the aim. This is a course on this topic. The aim, if you are those who are interested can obviously check various research articles in this. We cannot do justice to this in this particular class. Okay. So, and there are multiple recurrent loops within the cerebellum. What are these? What are the various recurrent loops within the or between the cerebellum and other parts of the. So, first this is, so it receives input from the cortex. So, the cerebellum receives input from the cerebral cortex via the pons, and then, so basically it goes via the cerebellar cortex and via the deep nuclei of, what are the deep nuclei? We said that the deep nuclei, these are the dentate, interposed and fastigial nuclei. These nuclei are the major output structures of the cerebellum. Via the deep nuclei they project to the ventrolateral thalamus, and from the thalamus to the primary and premotor cortex, thus controlling movement. So, that completes one loop, right, from the cerebral cortex back to the motor cortex, right. So, that is one loop. What is also there? What is also there is the inputs that are received from other regions. For example, we will take one case of vestibular input that is received. So, vestibular input serves as an information about head orientation and the position of the head, right. So, in other words, it is useful in maintenance of balance and posture, right. If this is somehow, if there is a disturbance somehow, that is detected by the vestibular system and cerebellum. So, the vestibular system to the cerebellum as in the flocculonodular lobe, and back to the vestibular nuclei and further into the vestibulospinal tract, right. So, here once again you have a recurrent loop that starts from the vestibular system to the vestibular nuclei. It turns out that the vestibular nuclei are special in the sense that they act like deep nuclei of the cerebellum. Vestibular nuclei are also among the major output structures of the cerebellum. So, vestibular nuclei, although they are located outside the cerebellum, actually act functionally similar to the deep nuclei of the cerebellum, acting as the output structure of the cerebellum. Okay. And also the case of the information that is received about balance and posture and maybe visual information, this is received in the vermal and the paravermal area, right. So, or the intermediate area of the cerebellum that projects to the, again goes through the cerebellar cortex, and then it projects to the limb extensors and axial musculature via the fastigial nucleus. The output in this case happens to be the fastigial nucleus. Once again the input that is shown here. So, once again the input is coming from multiple sources, but the output is on the limb extensors and more axial musculature for the maintenance of balance and posture. Okay. So, in this class we have seen that the climbing and mossy fibers encode different types of inputs, and the excitatory and inhibitory signals are classified by multiple parallel pathways. We have seen these, we have discussed some of these, as in the first excitation from the mossy and climbing fiber system is to the deep cerebellar nuclei, and mossy system further excites granule cells, and which excites the Purkinje cells in a divergent convergent manner, whereas the climbing fiber directly and with greater influence excites the Purkinje cell, and the Purkinje cells inhibit the deep cerebellar nucleus. So, there is a comparison of excitation and inhibition that is happening, and there are several recurrent loops that happen between the cerebellum and other regions of the brain. So, we will continue the discussion on cerebellum in future classes. So, thank you very much for your attention.